DNA extraction by non-organic method. In case of non-organic method, no organic chemical is used like the phenol or the chloroform. DNA is precipitated by inorganic salts by the higher concentration of inorganic salts like the sodium chloride. But there are some problems with this type of the DNA extraction, like if amounts of the salts they are left with the DNA, they would create difficulty for running on the PCR or as well as for performing RFLPs. Here is the procedure that how DNA is extracted with the help of non-organic method. During this procedure, the first step is cell lysis buffer. With the help of this buffer, the cell membranes of the cells of the blood sample, they are broken, while the nuclei, they are intact. So, as a result of first step, we are left with the pellet of the nuclei. This process is done by adding the cell lysis buffer and then washing the blood samples. Once we are left with the nuclei pellets, then these nuclei pellets, they are resuspended for the protein lysis buffer containing a high concentration of proteinase K. So there are nuclei which were present in the white blood cells, and there is present a protein lysis buffer, and there is a concentration of the proteinase K. As well as addition of the proteinase K, there is also added SDS in small amounts. During the step 3, this process, that is the addition of the proteinase K, SDS and protein lysis buffer, they lyse the nuclear membrane. Once the nuclear membrane is lysed or nuclear membrane is broken, this process is completed at a temperature 65 degrees centigrade. If the temperature is 65 degrees centigrade, then it is recommended for 2 to 3 hours. But if a person want to keep the sample on 37 degrees centigrade, then it is recommended for the overnight. So this process either can be completed within 2 to 3 hours or it can be kept for the overnight. If a person have to keep samples for overnight, then the temperature of the tubes should be 37, and if a person want to digest the proteins within a few hours, then she has to put the sample at 65 degrees centigrade for 2 to 3 hours. So during the step 4, temperature that denatures the protein and digestion of the protein takes place by proteinase K. Proteinase K is the enzyme which is used to digest the proteins. When we keep the sample at the temperature, and temperature or the SDS denatures the protein, and those proteins, they are digested with the help of enzyme. The name of the enzyme is proteinase K. During the next step, all those proteins which has been digested, they are removed from the DNA. So those proteins, they are removed with the help of an non-organic compound that can be lithium chloride or it can be sodium chloride. So supersaturated solution, mostly it is used sodium chloride. So the supersaturated solution is added to the digested proteins and it is incubated on the ice for few minutes. Proteins are precipitated out with the help of centrifugation. Once it is kept on the ice, then the proteins, they are precipitated by the centrifugation. DNA remains in the solution while the proteins, they settle down at the bottom of the tubes. So the DNA which is present in the solution is transferred to a new tube. Care must be taken so that there should not be amount of the protein transferred to the next tube. During the next step, DNA is precipitated by adding the isopropanol or the ethanol. So it can be precipitated either by adding the isopropanol or it can be precipitated by adding the ethanol. During the ninth step, precipitated DNA is washed with the help of 70% ethanol, dried under vacuum or in the room temperature, and it is suspended in the TE buffer. So this is the whole procedure of the purification of the DNA by inorganic method. In case of inorganic method, we use inorganic compound, that mostly NaCl. So with the help of NaCl, proteins, they are precipitated by performing the centrifugation. As a result of centrifugation, proteins, they settle down at the bottom of the tube and DNA remains in the supernatant. Supernatant is transferred to the next tube and amount of ethanol or isopropanol is added that precipitates the DNA. Once DNA is extracted or it is precipitated, it is washed with the 70% ethanol and it is suspended or dissolved in TE buffer for further use of the molecular biology. So in case of non-organic method, there is no use of the organic compounds. Usually this procedure is completed with the help of inorganic compound, and that is NaCl, sodium chloride. Digested proteins, they are removed by high concentration of salts like NaCl.